Sorry guys, because of the memory and the video has been stopped at the end. This is the continuation for the previous video. So this is how the in the same way how the electricity is produced, the hydroelectricity is produced. In the same way, the ATP is also synthesized when the protons will flow, and the protons will float through the ATP synthase complex. So it will flow through the F4 subunit. So these F4 subunits exactly it will flow to the C subunits of the F4 subunits. When the protons are flowing with high speed, the CO subunits will rotate. Okay, so to this CO subunit, this F4 this F1 subunit is attached. Okay, to this actually F1 subunit, this gamma handle is attached. This gamma handle also, when the C subunit and is rotating, C subunit is rotating, ultimately to lead to rotate this gamma subunit also, gamma handle also. When this gamma handle rotate, it will lead to some conformation in the B subunit. So this is actually called as binding change conformation, okay, in the beta subunit. Right, so binding change conformation, it's responsible for the synthesis of the ATP. Understood how the ATP is synthesized? So when proton gradient is generated in the intermembrane space, so due to the proton gradient, the protons try to enter into the inner mitochondrial membrane, but the inner mitochondrial membrane is impermeable, very much impermeable to the protons. So only way to enter into the inner mitochondrial membrane is the ATP synthase complex, that is complex 5. So when the protons are entering through the ATP synthase complex, it will enter to the subunits which are attached to which are present there. So it will first enter to the F4 subunit, especially to the C subunit. It will flow through the C subunit, 10 C subunits. When they flow through the C subunits, the C subunits will rotate. So this will leads to the rotation of the gamma handle. Okay, gamma handle which is present in the F1 subunit. Right, this when this gamma subunit rotate, it will lead to cause some conformation change in the B subunit, that is called actually binding change conformation. This ultimately leads to synthesis of ATP. Okay, this is the thing what I am going to I explained till now. Right, please concentrate on it so that you will understand. Right, sorry. So this is a total I explained previously. So in the complex 1, so NADH, from NADH the electrons will flow. So while electrons are flowing, so from the proton pump, the protons will be pushed into the intermitochondrial membrane space, in the intermembrane space. So from complex 2, it is not supplying any electrons. From complex 3, it is supplying 4 protons, and complex 4, it is supplying 2 protons. Total 10 protons are situated. So the positive energy, positive will increase here. Okay, but whatever the inner membrane it is there, it is impermeable to the protons. Okay, this protons, this inner membrane cannot allow these protons to enter. The only way is this complex 5. Okay, this is actually called as ATP synthase complex. Okay, so when these protons are flowing here, when the electrons are flowing here, sorry, not electrons, protons are flowing here, it has to flow through this subunits. Okay, this is what I am going to explain here. It will flow through the subunits. This is actually FO subunit. This FO subunit is total occupy or span this inner membrane. Okay, it will occupy total inner membrane. Right. So when the protons are flowing, like this, it is flowing through these subunits. This is called C subunits, not CO. I'm sorry. This is actually C subunits. When it is flowing to to this subunits, it will flow through this C subunit, and after that it will touch this F1 subunit. So this F1 subunit, it contains gamma handle and all the alpha beta subunits. After that, when it is flowing with a high speed, it will rotate C subunit and also it will rotate this gamma handle. So when this gamma handle get rotates, it will rotate the beta subunits, which will cause the conformation change. Okay, here I have written that F1 subunit attached to the F4 subunit. When F4 subunit leads to rotation of the F1 subunit, leads to rotation of the gamma handle. When this gamma handle will rotate, this cause actually what that is called binding change conformation. This cause conformation change to the beta units attached to the F1 subunits. Right, this ultimately will cause binding change conformation. This ultimately will leads to the binding change conformation. This leads to synthesis of ATP. Finally, NADH will synthesize 2 ATPs and when FADH2 synthesis 1.5 ATP. But why this is the thing? Why this will happen? Why NADH will synthesize 2.5 ATP and why FADH2 synthesis only 1.5 ATP? So when we look at the complex 1, okay, when we look at the complex 1, this is the complex 1. NADH is supplying how many protons? 4. This 4 and here total 10 protons. 10 protons, 4 protons will form 1 ATP. 4 protons will form 1 ATP. So 10 protons will form 2.5 ATP. So when we see complex 2, it is skipping complex 1. So it is missing 4 protons. So when it is missing 4 protons, so 1 ATP is missing. So 1.5 ATP it will form. So it is 4 protons, 1 ATP and 2 protons, 0.5 ATP. So 1.5 proton it will form. That is why NADH will form 2.5 protons and ATP will form 1.5 proton. Okay, this is the complex, right? Next slide. Now we will discuss about oxidative phosphorylation. What is oxidative phosphorylation? So it is very simple. When ATP synthesis is linked with the electron transport chain reaction, that is called actually oxidative phosphorylation. So the substrates first will get oxidized, then it will synthesize ATP. That process is called oxidative phosphorylation. Okay, when ATP synthesis is not linked with the electron transport chain reaction, that is called actually substrate level phosphorylation. Oxidative phosphorylation, ATP synthesis linked with the electron transportation. First, oxidation of substrates will take place, followed by the synthesis of ATP. Okay, when the ATP synthesis is linked with the electron transport chain reaction, that process is called oxidative phosphorylation. Okay, right. In another way, it involves actually PO ratio. What is P? Phosphate. Okay, what is O? Oxygen. Number of inorganic phosphate consumes and number of oxygen molecule consumes per ATP. Okay, it deals how many oxygen molecules are consumed and how many phosphate groups are consumed for ATP production, for one ATP production. That is deal by oxidative phosphorylation. Understood? In simple, it tells about how many ATPs are synthesized by the NADH and how many ATPs are synthesized by the FAD. Right? So I already told it, NADH it will form 2.5 ATPs and FADH2 it will form 1.5 ATP because NADH it will it is linked with the three complexes where total 10 protons are pumped. So FADH it is linked with the complex 3 and 4. It is not linked with the complex 1. So 1 ATP is less, right? So what are the sites of oxidative phosphorylations? There are I already discussed, right? There are 3 sites. Okay, Um, why these three sets? Because these are only complexes which will supply the protons. But complex 2, it is the oxidative phosphorylation will not take place in the complex 2 because it is not supplying any protons, right? This is the thing. So when we talk about when we talk about oxidative phosphorylation, we have to talk about the ATP synthesis also. We need to discuss about how um, so which complex will pump how many protons, how the protons are flowing towards the ATP synthesis complex. So how it is changing in conformation change in the beta subunits, and what is that called? It is called as binding change conformation. How that leads to formation of ATP, that we have to explain in the oxidative phosphorylation. Okay, since we have already explained the ETC, now here we are not going to explain the oxidative phosphorylation. Understood? So now energy formation in the metabolism. So in two ways the energy will be formed. One is actually substrate level phosphorylation, and another one is biological oxidation. So we already discussed about the biological oxidation. It will include ETC and oxidative phosphorylation, both oxidative phosphorylation, both it involve biological oxidation, right? But what is substrate level phosphorylation? So this is actually production of ATP while converting from one substrate to another substrate. During this process, ADP will use the inorganic phosphate to produce the ATP. This is the thing. Okay, it is not linked with the electron transport chain reaction. When one substrate is converting to another substrate, energy will be produced. How? The ADP, whatever it is formed, it will utilize the inorganic phosphate which is generating in the conversion, generating in the conversion. That inorganic phosphate, it is utilized by the ADP, then it will form the ATP. Okay, this is actually called as substrate level phosphorylations. So production of ATP during the conversion of one substrate to another substrate, this process is called substrate level phosphorylations. Here actually there is no necessity to go into the ETC, okay, that which is taking place in the mitochondria. Okay, substrate level phosphorylation need not to enter mitochondria to produce energy. Okay, this substrate level phosphorylation will takes place only in the cytoplasm. Okay, during the conversion of one substrate to another substrate, the energy is produced. Okay, this is called substrate level phosphorylation. Many examples are there where phosphoenol pyruvate is converted to pyruvate kinase is required here. This will take the inorganic phosphate from phosphoenol pyruvate and supply it to the ADP and forms ATP. Okay, here the energy is released, and the energy is released during conversion of one substrate to another substrate. So this is called substrate level phosphorylation. Okay, whatever the phosphate released by the phosphoenol pyruvate is utilized by the ADP to form ATP. This process is called substrate level phosphorylation, right? Next we will see the inhibitors. Okay, non-couplers of ETC and oxidative phosphorylation. There are a few inhibitors of complex 1. Okay, complex 1, there is pyrazine, guanidine, rotenone, it is a rat poison, and barbiturate, these are actually antidepressants. Okay, these are the inhibitors of complex 1. Like complex 2, melanoate, carboxyl, TIFA, that is a trienoyl trifluoroacetone. Complex 3, antimycin, Betis antileucide, and fenformin. Okay, these are the inhibitors of complex 3. Complex 4, actually carbon monoxide, arginine, cyanide, hydrogen sulfide. These are the inhibitors of the complex 4. And what are the inhibitors of oxidative phosphorylation? Atracitoside, oligomycin, oxidative phosphorylation, and we can call it as inhibitors of ATP synthase complex also. Potassium ionophores, this also inhibitors of ATP synthase complex or complex 5 or oxidative phosphorylation. Okay, here we will use varenomycin, gramicidine. These two also inhibit the complex 5 or ATP synthase complex. Okay, now we will enter into the uncouplers. There are two types of uncouplers. One is physiological uncouplers and chemical uncouplers. What is the mechanism of uncouplers? Very simple. This actually increase the permeability of protons, so no proton gradient will be synthesized, right? No proton gradient will be synthesized, so no ATP synthesis will be there. So it will increase the permeability of the inner mitochondrial membrane. When the permeability of the inner mitochondrial membrane will increase, so the protons will enter into the intermitochondria, inside the mitochondria, so there will be no proton gradient. So when there is no proton gradient, what will happen? The protons cannot flow towards the ATP synthase complex, and there will be no ATP synthesis. That's it. Okay, this is very simple. And physiological uncouplers are thyroxine, long chain fatty acids, unconjugated bilirubin, and thermogenin. Okay, next chemical uncouplers are 2,4-dinitrophenol, 2,4-dinitrocresols. Okay, these are the examples, few examples. And aspirin high dose also would be used as a chemical uncouplers, and and calcium. These are the uncouplers, right? Next, I need to explain little bit about the little bit about the what ATP synthase complex. This is actually figure of ATP synthase complex. So here actually proton gradient is established in the intermembrane space. Okay, so this side actually outer membrane will be there. So when the gradient is forming, so the protons try to push into the intermembrane, inside the membrane, but unfortunately this membrane do not allow, inner membrane, mitochondrial membrane do not allow, because this is impermeable to the protons. Okay, because of this proton gradient will be formed in the intermembrane space, and there will be positive here and there will be negative inside the membrane, that means in the matrix. Okay, use the word matrix, that will be better. Okay, so the protons try to enter into the matrix, but this inner membrane cannot allow, because this is impermeable. So the only way is that it will pass through the ATP synthase complex. This is F4 subunit, okay, which is attached with the C subunits. There are 10 C subunits will be there. So when the protons are flowing, this will rotate like this. F4 subunits along with the C subunits will rotate. When this is rotating, this F1 subunit also rotate. So whatever the handle, this is called gamma handle, this will rotate along with this subunits also rotate. This will lead to develop a conformation change in the beta subunit. Okay, this is called actually binding change conformation. This will lead to synthesis of ATP. Okay, thank you guys. This is overall electron transport chain. Thank you for having patience. Okay, have a nice day.